Concept number 2. Let's have a look at prokaryotic and eukaryotic cells. All cells share certain basic features. They are all bounded by a selective barrier called the plasma membrane, also referred to as the cell membrane. Inside all cells is a semi-fluid, jelly-like substance called cytosol, in which subcellular components are suspended. All cells contain chromosomes, which carry genes in the form of DNA, and all cells have ribosomes, tiny complexes that make proteins according to instructions from the genes. A major difference between prokaryotic and eukaryotic cells is the location of the DNA. In a eukaryotic cell, most of the DNA is in an organelle called the nucleus, which is bounded by a double membrane. In a prokaryotic cell, the DNA is concentrated in a region that is not membrane enclosed, called the nucleoid. The interior of a prokaryotic cell is called the cytoplasm. This term is also used for the region between the nucleus and the plasma membrane of a eukaryotic cell as well. Eukaryotic cells are generally much larger than prokaryotic cells. Size is a general aspect of cell structure that relates to function. The logistics of carrying out cellular metabolism sets limits on cell size. So here we have a nice picture or a nice figure of a typical rod-shaped bacterium. And you can see we have the fimbriae. These are like attachment structures on the surface of some prokaryotes. Then we have the nucleoid. This is where all the DNA is located. We have the ribosomes. This is where all the proteins are made. We have the plasma membrane. This encloses the cytoplasm. On the other side of the plasma membrane, we have a rigid structure outside the plasma membrane known as the cell wall. And then we have this like capsule or jelly-like outer coating of many prokaryotes. And finally, at the end of the rod, we have a flagella. This is used for movement and translocation of the bacteria. Now, at the boundary of every eukaryotic cell, there is the plasma membrane. This functions as a selective barrier that allows passage of oxygen, nutrients, and wastes to service the entire cell. Now, for each square micrometer of membrane, only a limited amount of a particular substance can cross per second. So the ratio of surface area to volume is critical. As a cell or any other object increases in size, its volume grows proportionally more than its surface area. Area is proportional to a linear dimension squared, whereas volume is proportional to a linear dimension cubed. Thus, a smaller object has a greater ratio of surface area to volume. So in this slide, we can see the geometric relationships between surface area and volume. Here, the cells are represented as boxes. Now, using arbitrary units of length, we can calculate the cell's surface area in square units or units squared, volume in cubic units or units cubed, and the ratio of surface area to volume. A high surface to volume ratio facilitates the exchange of materials between a cell and its environment. Now here we have a nice figure showing the plasma membrane. Now the plasma membrane and the membranes of organelles consist of a double layer, a bilayer of phospholipids with various proteins attached to or embedded in it. Inside or the interior of a membrane, the phospholipid tails are hydrophobic, as are the interior portions of membrane proteins in contact with them. The phospholipid heads, or the top of the phospholipids, are hydrophilic, as are proteins or parts of proteins in contact with the aqueous solution or the water solution on either side of the membrane. Carbohydrate side chains, you can see on the top of the membrane, are found only attached to proteins or lipids on the outer surface of the plasma membrane. So here we have a nice panoramic view of the eukaryotic cell or an animal cell. Now, in addition to the plasma membrane and its outer surface, a eukaryotic cell has extensive and elaborately arranged internal membranes which divide the cell into compartments. The cell's compartments provide different local environments that facilitate specific metabolic functions. So, incompatible processes can go on simultaneously inside a single cell. Now, animal cells, believe it or not, have much more in common with plant cells rather than prokaryotic cells. Now, if we look at more detail the function of each of these compartments, we can see that the endoplasmic reticulum, or ER, consists of rough ER and smooth ER. And these are essentially membranous sacs and tubes, which are active in membrane synthesis and other synthetic and metabolic processes. The nuclear envelope is essentially a double membrane enclosing the nucleus, perforated by pores. The nucleolus is a structure involved in the production of ribosomes. A nucleus has one or more nucleoli. And then we have the chromatin. This is a material consisting of DNA and proteins, visible as individual chromosomes in a dividing cell. Now, let's look at two cellular components involved in the genetic control of the cell: the nucleus, which houses most of the cell's DNA, and the ribosomes, which use information from the DNA to make proteins. The nucleus contains most of the genes in the eukaryotic cell. Some genes, however, are located in the mitochondria, and in plant cells are located in the chloroplasts. Now, the nucleus is generally the most conspicuous organelle in a eukaryotic cell. It averages about 5 to 6 micrometers in diameter. Now, around the nucleus, or enclosing the nucleus, is the nuclear envelope. Now, the nuclear envelope is a double membrane, essentially two membranes. Each has a lipid bilayer associated with proteins, and these bilayers are separated by a space of about 20 to 40 nanometers. The envelope itself is perforated by pore structures that are about 100 nanometers in diameter. So here we have a nice picture of the nucleus and its envelope. Within the nucleus are the chromosomes, which appear as a mass of chromatin, and one or more nucleoli, which function in ribosome synthesis. The nuclear envelope, which consists of two membranes, is separated by a narrow space and is perforated with pores and lined by the nuclear lamina. Now, the nuclear envelope is perforated or contains pore-like structures that are about 100 nanometers in diameter. At the lip of each pore, the inner and outer membranes of the nuclear envelope are continuous. Now, an intricate protein structure called a pore complex lines each pore and plays an important role in the cell by regulating the entry and exit of most proteins and RNAs, as well as large complexes of a macromolecule. Except at the pores, the nuclear side of the envelope is lined by the nuclear lamina. This is a net-like array of protein filaments that maintains the shape of the nucleus by mechanically supporting the nuclear envelope. So what is the nuclear lamina? The lamina is a net-like array of protein filaments that maintains the shape of the nucleus by mechanically supporting the nuclear envelope. Now, within the nucleus, DNA consists of a separate unit called a chromosome. The chromosome carries the genetic material. Each chromosome is made up of a material called chromatin. This is a complex of proteins and DNA. As the cell prepares to divide, thin chromatin fibers coil up or condense, becoming thick enough to be distinguished as the familiar separate structures we know as chromosomes. A prominent structure within the non-dividing nucleus is the nucleolus, which appears through the electron microscope as a mass of densely staying granules and fibers adjoining part of the chromatin. Here, a type of RNA called ribosomal RNA, or rRNA, is synthesized from instructions in the DNA. Also, in the nucleolus, proteins imported from the cytoplasm are assembled with rRNA into large and small ribosomal units. These subunits then exit the nucleolus through the nuclear pores to the cytoplasm, where a large and small subunit can assemble into a ribosome. Sometimes there are 2 or more nucleoli. The number depends on the species and the stage in the cell's reproductive cycle. Now, the nucleus directs protein synthesis by synthesizing messenger RNA, or mRNA, according to instructions provided by the DNA. The mRNA is then transported to the cytoplasm via the nuclear pores. Once an mRNA molecule reaches the cytoplasm, ribosomes translate the mRNA's genetic message into the primary structure for a specific polypeptide. Now, ribosomes, which are complexes made of ribosomal RNA and protein, are the cellular components that carry out protein synthesis.
Cells that have high rates of protein synthesis have particularly large numbers of ribosomes as well as prominent nucleoli, which makes sense given the role of nucleoli in ribosome assembly. For example, a human pancreas cell, which makes many digestive enzymes, has a few million ribosomes.
Now, ribosomes build proteins in 2 places, or 2 cytoplasmic locales. So at any given time, free ribosomes are suspended in the cytosol, while bound ribosomes are attached to the outer side of the endoplasmic reticulum or nuclear envelope. Bound and free ribosomes are structurally identical, and ribosomes can alternate between the 2 roles. Now, most of the proteins made on free ribosomes function within the cytosol. Examples are enzymes that catalyze the first steps of sugar breakdown. Bound ribosomes generally make proteins that are destined for insertion into membranes, for packaging within certain organelles such as lysosomes. Cells that specialize in protein secretion, for instance the cells of the pancreas that secrete digestive enzymes, frequently have a high proportion of bound ribosomes.